then, I mean, first of all, thanks for the very kind introduction, Rodrigo. It's a pleasure to be here and present our research. And I'm not only excited because this is actually my first invited seminar, but also because we have some really cool stuff to present here. So I'm very excited to hear what you think about it. So none of this is currently published, and to some degree it's still in progress. So please forgive me if one or the other thing are not as clear yet as they will be eventually. Okay, so title of the talk: a theory for Hebbian learning in recurrent IA networks. So when you read learning in networks in one sentence, most people these days probably think of deep artificial neural networks that are trained with backpropagation. And rightfully so, because these networks have demonstrated that they can perform on a sometimes superhuman level on many interesting tasks, such as, for example, object recognition. Now, this here is an example how such a network looks like. So you feed in an image here on the left-hand side, then you have multiple processing layers, and at the end you collect a label for this network. And what is interesting is that recently people have found out that the better these networks perform on the image recognition tasks, the more brain-like their activity seems to be. So what do I mean by that? In this experiment, the authors presented different images to a monkey while they recorded the brain activity along the ventral stream here, which is thought to be responsible for image recognition. And they were able to record these spike trains here with these microelectron arrays. And in a more recent study now, the same people took the data and, they fed in the same images here to these artificial neural networks and tested how well these networks can predict the activity in the monkey brain based on the activity of the artificial neurons in these different layers. And what they did is they matched these different layers according to the hierarchy of this processing stream as it is in the ventral stream of the monkey. And from these prediction accuracy, they computed what they call a brain score for these artificial neural networks. And when they plotted the brain score of the network and compared it to the performance of the network to a standardized image recognition task, what they observed is that the better the networks performed on these image recognition tasks, the higher their similarity to these brain recordings was, which is super interesting because this tells us that, I mean, what you might expect is that the brain can tell us a lot about image recognition. And so this can motivate why we should maybe take a closer look to, why we should try to understand what's going on in the brain, because this potentially yields a lot of performance improvements for artificial neural networks. So what are the main differences of artificial neural networks and brains? And there are probably a lot of things I could talk about now, but one of the main aspects is that we have much fewer of these processing stages here. So in this example neuron here, we have more than 10 of these stages. However, when we look at biological networks, what we rather have in terms of, for the specific task of object recognition, is that we probably have at most 6, if we take the retina into account here, 6 processing stages until we get a behavioral response as an output of, for example, a monkey performing a specific task. So, and this becomes even more complicated when you zoom in. So, for example, when we take now the V1 here, the primary visual cortex, as one of these processing stages, and look at what this looks like in the brain, we see that this processing stage itself is can be decomposed into multiple layers. And when we look at the connectivity in these layers, or in and between these layers, what we see is that there's a lot of recurrency. So, for example, in this case, this is data from primary visual cortex of cat. We see that 20% here of the excitatory connections in this cortical area are recurrent connections within this layer 2-3 of this cortical sheet. So it seems like recurrence is really one of the main differences between the brain and artificial neural networks. And we can zoom in even further into these layer 2-3, and what we see there is here sketched with this in this circuit diagram, that we have many different types of neurons. So in this case, what I show here is, on the one hand, excitatory cells indicated by these blue triangles, but also inhibitory cells indicated by these magenta circles. And this is already a super simplified depiction of what is actually going on, because we have actually many, many different types of interneurons, as these inhibitory cells are called. And they all are connected in a different fashion. And it's a whole zoo of subtypes that are very hard to make sense of. So the circuit diagram here is already a vastly simplification of what's actually going on. Okay, so with all this biological detail, when I say that we should be inspired by the brain with our artificial neural networks, it's of course the question, where do we start? I mean, there are so many aspects you can draw inspiration from. And what I want to do today is I want to focus on one interesting computation that can be performed by such a circuit that I show here on the right, and that is response normalization. So what is response normalization? Let me try to explain this to you in terms of an experiment. So in this study, the authors placed a microelectron array over the primary visual cortex of cat and binned the recording sites according to their preferred orientation tuning, which is indicated by the colors here. And what they then did is they presented different stimuli that were essentially different gratings that are oriented in different directions. So first they established a baseline response where none of the gratings here was presented, and this is the recording they observed. And then they presented one of the gratings at 50% contrast, in this case here the horizontal grating, and then they observed a response of the neurons that were selective for this respective orientation. And they did the same for the other orientation, and they see a different set of neurons become active. Now, the interesting thing is when both of these gratings were presented simultaneously at 50% contrast, here on the bottom right, which resulted in this checkerboard pattern-like stimulus, the response was normalized, meaning that the peaks were much lower than in the single grating trials. And this is different from what you might naively expect, namely that the responses of the combined gratings is the sum of the responses to the single gratings. Okay, so you have some kind of normalization of these responses. And I mean, if you were doing machine learning and deep learning in particular, you say, oh, this is nothing new. We are doing this all the time across layers, across regions, and across features. But let me try to convince you that here's something more complicated going on. So what they also did is, in the study, they presented now this one of the stimuli at a lower contrast level. And again, presented it alone and observed that actually the response magnitude here is very similar, independent of the contrast level of the grating. So this again could be just a scaling factor, right? But now the interesting part is when they presented both of the stimuli together on the bottom right-hand side, which resulted also in a similar checkerboard pattern as before, but now you have the slight preference for this horizontal grating because it is presented at a higher contrast. What is observed is that this weaker stimulus is strongly suppressed by the network. And again, you might say, oh, this is just some softmax function that we apply to the cell, but it's not entirely clear what to optimize the softmax for, because in terms of pixel distance, this stimulus here that was presented has the same distance to this purely vertical presented grating and to the previously presented checkerboard grating. So it is not clear which of these features are actually interesting to the animal. And it seems like in this particular case, the cat has a strong prior for these pure gratings. And it's not clear where these priors come from and how, especially how they are learned. And so one computational model that can explain some of this is the Stabilized Supralinear Network that Rodrigo already mentioned. So let me briefly introduce to you what this is all about. So the Stabilized Supralinear Network, or in short SSN, is a theoretical model of inhibitory and excitatory cells here in blue and magenta. And these cells here are recurrently connected with each other, as is observed in these cortical layers. And they are stabilized by inhibition, meaning that, the excitatory cells here are stabilized by inhibition, meaning that without the inhibitory input, the activity would grow larger and larger due to these recurrent excitatory connections. And additionally, the network is supralinear in that each cell has a supralinear input-output mapping that follows a power law nonlinearity. you can think of the input axis here as a membrane potential of the cell, and the output axis here as the firing rate Z of the cell. And this is a nonlinear function. Now, in addition to these recurrent connections, each cell also receives feedforward input that is tuned to a specific orientation. So when we pick out these two cells here that are tuned to an angle of around 40 degrees, we can assign them a sort of a tuning curve. And when we present now a particular orientation theta to the network, the crossing point between the tuning curve and this specific dashed line here indicate, so the y coordinate of this point here gives the input current that is fed feedforwardly to both of these cells. And it's to both of these cells because here inhibition and excitation in this simplified scenario have the same orientation tuning. And now with the setup combined with we can of course do this now for all the cells and not only for two of them. And then we have this nicely distributed tuning curves across the whole stimulus space. And so from this procedure here now, from all these crossing points, we can now calculate the input that is fed into these networks, into these neurons. And together with the recurrent connections, eventually the network settles down, and we can read out the response of the network by the excitatory firing rates of these different cells that are tuned to different orientations. And as you see here, when we present this particular stimulus, we see that also cells that are not exactly tuned to the stimulus are slightly active. So the orange dots here are the respective cells. Okay, so with this setup, we can now essentially reproduce the experiment that I just explained to you. So we first present one stimulus indicated here by this orange response curve, which is exactly the same as the bottom left here that I just explained to you. Then we can present a second stimulus here in green, which corresponds to a different grating essentially. And then we can present them both together. And each time we read out the firing rate of the excitatory cell population and plot them according to their preferred orientation here. And this, what we find out, or what we see here, is exactly what I just described to you in the experiments, that the response to the combined stimulus here is actually below the response to the single presented grating. And this, of course, also works when we change the input magnitude of the different gratings, which would correspond to the different contrast levels. So in this case here, the orange stimulus has a 3 times larger contrast than the green stimulus. And as in the experimental data, we see that the weaker stimulus here is strongly suppressed. And the network really favors this orange stimulus that is much stronger in the input. Okay, so one major drawback of this network is that all the weights here are static. So they are taken from experimental data, of course, and therefore we probably also, this is the reason why we see the same kind of normalization. But it would, of course, be cool to learn all these connections from the raw stimulus statistics. And in the following, I want to show you how you can actually do this. You can learn all these connections using a biologically plausible plasticity rule, namely Hebbian learning. And as a first step, I want to focus on the connectivity between inhibitory and excitatory cells and how they are balanced in the brain. So from experiments, we actually know that excitatory and inhibitory inputs to a single cell are balanced. So in this particular study, these are results from rat somatosensory cortex, where they did patch clamp recordings while the network was spontaneously active. And what you observe is that both inhibitory and inhibition and excitation were highly symmetric in the activity, in the sense that when there was a lot of excitatory input, as for example, here in this peak, there was also a lot of balancing inhibition coming in, as you see in the blue curve here. And the same is actually true in the case of evoked activity. So in this case, they recorded from synaptic input to a neuron in rat auditory cortex while they played tones of different frequencies. And what you see is that the inhibitory and the excitatory tuning curves are very closely matched. So in blue, you see that the inhibitory tuning curve is slightly broader than the excitatory tuning curve here in red. And of course, the question is, how can we, this is a very, very, very general phenomena. So This balance of inhibition and excitation is observed across many different brain regions and species. So this is really a very defining property. And the question, of course, is how can we learn this with a biological rule? And for this, let me briefly introduce to you what Hebbian learning actually is. So Hebbian learning can be summarized in one sentence as cells that fire together, wire together. And this means when we have two cells here, one presynaptic cell Y and one postsynaptic cell Z that are connected with a synaptic weight W, then the plasticity or the change of the synaptic weight here is proportional to the pre times the postsynaptic activity. In this case, this is equal to the presynaptic to Y times Z, where you can think of Y and Z as just firing rates of the neuron. And so what this means is that when you have a presynaptic spike train happening here in the cell, and that triggers a postsynaptic spike train in the postsynaptic cell, this leads to an increase of the synaptic weight. So this is this learning rule is in itself interesting because it is experimentally very well established. But on the other hand, it's also interesting when you are maybe interested in neuromorphic hardware, because this learning rule is totally unsupervised and local in its sense, because it only requires the pre and postsynaptic activity, and in that sense we have no error signal or anything. So potentially there's a huge potential to apply this rule in neuromorphic hardware. So however, there's one problem with this learning rule, and that is when due to the increase of the synaptic weight, the presynaptic neuron may trigger additional postsynaptic spikes in the postsynaptic cell, which would lead to additional plasticity again, and even more spikes potentially in the postsynaptic cell, and so on and so on. So essentially, we have an unlimited growth of this this connection here. And when we introduce recurrency, this will get only worse. So in order to understand this better, let us first phrase all of this in a few mathematical equations. So now we have a slightly more complicated model where we have a set of input cells Y, where Y here is a vector now, and that are connected with a via synapses W to the postsynaptic cell Z. And we consider the Z here being the output firing rate of the postsynaptic cell. And in this case, Z is a linear function of the input, namely the dot product between the input cells Y and the synaptic weights W. And let me briefly mention here, most of our theory that we derive only works in the case of a linear activation function. However, from this numerical simulations, we see that a lot of what we understand of the linear system carries over to the nonlinear case, where we have a power law nonlinearity here, as in the SSN. So, however, when we stick with the linear output function here for a second, what we observe is that the expected synaptic change here is proportional to the covariance matrix times the weight as it is currently. And so the average here is over multiple training sets. And for better readability, I will drop these angled brackets in the future. So this, of course, is a very simple differential equation, and we can solve this in the eigenspace of the covariance matrix C. And what we figure out is that essentially we have unlimited growth along these eigencomponents of the weight, where the growth strength is governed by the eigenvalues of the respective eigenvectors. So when you think of this point cloud here of your data, so this is just getting the first two components here of your inputs. What we see is that we grow along these principal components here with the strength that is proportional to the eigenvalues. Okay, so the question is, how do we actually stabilize this unlimited growth? And one hypothesis how this plasticity can be stabilized is the competition for synaptic resources within a dendritic branch. So this is motivated by experiments done by Bourne and Harris in 2011 in hippocampus, where they induced theta burst long-term potentiation, LTP, and compared a reconstructed part of the dendritic branch before and after the LTP induction. And what they found is, while the synapses here become much bigger, the average synaptic area per dendritic length here was maintained. So what does this mean for our plasticity rule? Essentially, when the total synaptic area is maintained, this corresponds to a normalization constraint for our synaptic weights. And this was already figured out a few decades ago. When we have this constraint here, we can build this into our update rule with this additional subtractive term. And now what we have is essentially an eigenvector equation for the fixed points of the system. So when we want to calculate the fixed points, we have to set our W dot here to 0. And what we get is the eigenvectors of the covariance matrix as fixed points. And eigenvectors of covariance matrices are usually the principal components. And this is exactly what this neuron here performs in this linear learning setting. Okay, and in terms of when you're more used to these kind of point cloud plots, when we have our data here as this point cloud, then the weight vector aligns with the principal most dominant eigencomponent of the covariance matrix. Okay, so this was already very well established. Now, what we added to the story is that we realized that this normalization is not only happening for excitatory weights, but actually also for inhibitory weights. So again, when you look at the total synaptic area before and after the induction, this is maintained. So essentially, we can do exactly the same equations, but now we just have this additional constraint and implement this into our update rule. We get a slightly different matrix, which was before the covariance matrix. So this is no longer a covariance. Therefore, we call it a pseudo-covariance matrix, C prime. But the principle holds that we can calculate the fixed points as the eigenvectors of this matrix. All right, so this is very abstract, right? I mean, maybe some of you have an idea what these eigenvectors and eigenvalues mean and what they look like, but let me show you an example of what it actually means that the principal eigenvectors of these covariance matrices are stable fixed points. And for this, we make a simplifying assumption, and that is that we have the inhibitory and the excitatory inputs here to our postsynaptic cell. We set them equal. And this is motivated by the fact that actually in the cortex of, in the auditory cortex of rat or in cat visual cortex, we observe that the tuning of inhibitory and excitatory cells is similar in their sharpness. So therefore, we can make this assumption, and then suddenly this C prime matrix here becomes a lot simpler, and we can actually immediately read out the fixed points of the system, or better to say the eigenvectors of the system, which are the fixed points of the weight dynamics. And what we see is that essentially we have a simple sign flip in the excitatory component of the vector and in the inhibitory component of the vector. So when we again look at this from the perspective of our input space, what we see is that as before, we do PCA for our excitatory component, and now the inhibitory component of our feed-forward weight vector here is goes just in the opposite direction. And so this all works for the linear case, but from numerical simulations, we can also do the same thing in a slightly more complex scenario where we have different input neurons, but now present different orientations to them. And when we let the system run, what we see is that we get these more complex tuning curves, but they are still, as in the linear case, nicely co-tuned. Okay, and this was not possible with the previous inhibitory rules. So we think that this is a first step to actually understand this on a more principled basis, the recurrency on a more principled basis. Okay, so the problem is that all that I told you right now is just feed forward, right? So, but eventually you want to learn a recurrent network, and this is, of course, much more complicated. But as a first step, what we tried is we added additional lateral input that is not plastic, but tuned to a specific eigenvector P of the feed-forward covariance matrix, shown here in light blue. And now what we ask is, how does the original learning dynamics for the original neuron here in dark blue, how is this affected by this static lateral input from this other light blue neuron? And essentially, mathematically, what we do is we essentially increase the dimensionality of our input space by this lateral component from this additional lateral excitatory cell. And what this leads to is that we have a new kind of covariance matrix of which we can again calculate the eigenvectors and the eigen directions, and eigenvalues. And what we find is that the one eigenvalue, the eigenvalue that corresponds to the one eigenvector that this additional lateral neuron here is tuned to, increases its attraction or increases its dominance. And again, this is very abstract. Let me try to explain to you this in a more visual way. So again, here we have in blue our original inputs, and now what I do is I increase the norm of this feed-forward weight vector of the static additional input. And what we see is that it effectively transforms our input space such that when we perform principal component analysis on this input space, so all of a sudden different directions can become dominant in their attraction. And when we, so when we then perform, go back to our nonlinear system here, what we did here is we introduced this additional input here just as a static tuning curve here, and then initialize the feed-forward weights here of the original neuron randomly. What we see is that this specific input node here is dominant in its attraction, and the other neuron becomes attracted to this direction. Okay, so this is what happens when we add a lateral excitatory input. And now when you add an additional inhibitory input, essentially the same thing happens. So let me go through this very quickly. Again, we have a different kind of covariance matrix. We have this additional input dimension. This time it's inhibitory. But now instead of increasing the attraction along this particular eigenvector, now we decrease it. And in this input space, what this corresponds to is that we, instead of stretching the input data, we now quench it together. And as you might intuitively guess, in this more complex input scenario, what happens is that we essentially introduce a repulsion of specific areas of the input space by adding multiple inhibitory cells here. So what we did here is we distributed these inhibitory cells across the similar space, but kept a small gap. And what you would expect is now is exactly what happens is that the one plastic excitatory cell here becomes selective for this gap, which is the only region of the input space that is not suppressed by these lateral inputs. Okay, and finally, of course, we can combine both of these results and state that the total attraction of an eigenvector is given by a feed-forward component, which is this first term here, and a lateral component where excitatory and inhibitory cells increase the attraction, and inhibitory cells decrease the attraction of this particular mode. Okay, this, but still, this is not a feed-forward. This is still in its core a feed-forward network, so we have no recurrent connections. But from this, we can build some intuition to better understand what's actually happening in the recurrent case. So this now is a fully recurrent network where we have two excitatory cells, and they are connected to themselves and to each other. And all of these connections are plastic, but what I show you here is only the feed-forward tuning curves of these cells. And the way we initialize them is that they are already tuned to different orientations with a small overlap in their receptive field. And what happens is that due to this small overlap here, the firing of these postsynaptic cells is correlated, which leads to an increase of these recurrent connections. And therefore, as before, they essentially attract each other and the tuning curves become clustered eventually. And this is, of course, not necessarily what we want, right? So when we have multiple cells, it would be maybe a good idea to have different orientations that we cover in the stimulus space. And interestingly, when we do the same thing for only inhibitory cells, there what we observe is that the tuning curves push each other apart, and due to this repulsion, the cells distribute the tuning nicely across the stimulus space. Okay, so the question is, how can we present the clustering of these excitatory tuning curves by somehow taking advantage of the decorrelation of these inhibitory cells? And the basic idea is that the increase of attraction due to this additional excitatory neuron here is balanced by a simultaneous decrease of attraction by an additional inhibitory neuron. And let me try to make this a bit more clear. All right, so now think of a network that is fully connected and also receives feed-forward input, which I do not show here to keep the picture a bit clearer. The problem is that we cannot solve this system because even in the case where we have linear activation functions, this is a nonlinear coupled differential equation that, at least in our hands, we weren't able to solve it. So, however, in the linear case, we can look at very specific states of the system and ask what the stability would look like and look for inconsistencies. So let me try to guide you through this step by step. So instead of just one additional lateral input, we can think of a case where we now have a whole bunch of neurons that are tuned to a specific feed-forward eigendirection of the circuit. And since we call this an eigencircuit, since all the neurons in the circuit are tuned to the same eigenvector of the feed-forward covariance matrix. So if this circuit is stable, the expected weight change must be 0 for each of these neurons. And in this local equilibrium, we can actually solve this and calculate now, as before, the attraction now of the whole eigencircuit as this terms here. So in this case, we have one term, again, that is just the feed-forward attraction of this particular eigendirection. And we have a modification due to the excitatory neurons and due to the inhibitory neurons. Now, the only difference to the non-recurrent case is that these variances here depend on the number of E and I cells in the eigencircuit and also on as before, on the weight norms, where the WEI here, for example, is the total synaptic inhibitory weight that each excitatory cell receives. And so we do not, for these weight norms, we do not distinguish between feed-forward and recurrent inputs. So what we now assume is that all these cells here can be assigned to one of these eigencircuits, meaning that they are tuned to one particular eigendirection of the feed-forward covariance matrix. And since these different eigenvectors are orthogonal to each other by definition, the activity of these neurons is uncorrelated. And the weights across these eigencircuits here in light gray, they decay to 0 eventually. So since we assumed that the system is in equilibrium, we can now write down the attraction, as I try to explain here on the right. We can write down the attraction for each of these single eigencircuits. And for this toy model here, let's assume that each excitatory cell here contributes +1 to the attraction, which would correspond to this term here, and each inhibitory cell contributes -1, and we just set this baseline here to 0. So actually, you can think of this as a set of charged particles that create a sort of a potential in this high-dimensional space. So when we do this, we can now plot the eigencircuit attraction of these different eigencircuits, and what we observe is that depending on how many inhibitory and excitatory cells you have, the eigencircuit is either attractive, as in for the third circuit here, or it can be repulsive, as for every other circuit. And we can again ask if this configuration is stable. And in fact, it is not, because neurons from the eigencircuits that are repulsive become attracted to the more attractive eigencircuit and change their tuning accordingly. So this is independent from neuron type, since every neuron, all excitatory and all inhibitory, see the same attraction landscape. So in this toy example, let's assume that all neurons in fact become attracted to this third eigencircuit. And what we can do then, again, think about what the attraction landscape would then look like, which I show here in these dashed lines. And now the previously dominant eigencircuit actually becomes repulsive. And the reason for that is that each increase in the attraction due to an additional excitatory cell here is actually balanced by a decrease of the attraction, since we also add 2 inhibitory neurons. And if we continue this procedure, in the sense that we again ask, so where do these cells go next, since they are in this repulsive eigencircuit, we see that they get pushed out of the circuit and then become redistributed across the stimulus space. So in general, we can prevent the collapse of these tuning curves if for every excitatory neuron, we have a sufficiently large fraction of inhibitory neurons that become attracted to the dominant eigencircuit. And we can formulate this in an equation that states that the change of the total attraction of an eigencircuit has to become negative, and where the fraction of the inhibitory cells is given by the total number of E and I cells in the system. So while this is just a toy example, and it only works for a linear network, however, it gives us a lot of intuition how we have to change these parameters to actually maintain this constraint and have a nonlinear system to decorrelate. And now let me show you what this looks like. So now here we have a fully plastic recurrent network that receives feed-forward input from a layer of excitatory cells here in faint blue. And on the left-hand side, I show you the feed-forward excitatory weights of all excitatory neurons and the feed-forward excitatory weights of all inhibitory neurons. And when we run the simulation, what we see is that, different from before, now also the excitatory neurons distribute across the stimulus space as we would like them to. And we can not only look at the feed-forward weights, but we can also look at the recurrent weights and what we observe here. So on the x-axis here, we sorted these neurons according to their tuning preference. And what we see is that here in the diagonal, we have the strongest synapses, which means that neurons are preferentially connected to similarly tuned neurons. And this is in very good agreement with experimental data across different species and sensory modalities. Okay, so this is about the weights, but we can of course also look at the actual firing rates and the actual activity of the network. And what I plotted here is the response tuning curve centered around the peak tuning orientation averaged over all cells of each cell class. So what we find is that inhibitory cells here in red are slightly broader tuned than the excitatory cells in blue. And this is also reflected in the input conductances. So this are now the conductances to the excitatory cell population. In red, the inhibitory input, and in blue, the excitatory input. And again, as for the firing rates, inhibition is slightly broader tuned. And as for the weights, this is in good agreement with experimental data. Okay, so we can learn these weights in a very nice way, and apparently this corresponds to what was actually happening in the brain, or at least the end result is what looks like what we see in the brain. So the next question is, can we do anything useful with these networks? So can we do some actual computations? And in fact, I wouldn't speak to you today if we couldn't. And essentially, our network is a stabilized supralinear network. So what we did is we reproduced the response normalization experiments that I explained to you already. And as for the SSN, what we observe is that we have this response normalization for the case we have the same contrast for both of the stimuli. And in the case where we have this more dominant stimulus, we have this strong suppression of this non-dominant stimulus. And for the experts, let me briefly mention that we also checked if our network can reach the inhibition stabilized regime. And we checked this by providing an additional stimulus to the inhibitory population and observed that their activity here in magenta paradoxically decreased in accordance with these theoretical and experimental results. Okay, so while these phenomena here were already known from the static stabilized supralinear network, all of this, let me stress that all of this now is learned from the raw stimulus statistics. And we have no hardwiring of any connections, and everything is plastic at the same time. Okay, so until now, everything I presented to you was already explained by the static SSN, and the only difference was that we can now learn these connections. But let me now come to some new results that we found in our network. So in a specific parameter regime, what we observe is that our tuning curves become clustered and decorrelated at the same time. So what do I mean by this? So in this particular simulation, what you see is that the tuning curves are at the beginning nicely distributed across the stimulus space, but then over time, you see that you get these tuning modes that form. So, and even though you have this significant overlap between different tuning curves, the system does not merge to a single dominant tuning curve. And so this is not due to the input stimulus statistics. What we actually have here is that we present different orientations on the x-axis with equal probability. So the input distribution here is uniform, and nevertheless, we see these peaks here in the tuning curves, or this clustering. And the question, of course, is how does this work, and where does this clustering come from? And what we think is that what's going on here is that is due to our input-output nonlinearity, as in the SSN, our network can be in both regimes at the same time. So on the one hand, the decorrelating regime, and on the other hand, the non-decorrelating regime. So as an example, when we drive the network here, exactly at an orientation that lies between these two tuning peaks, the neurons are only weakly activated. And when we linearize our system around this fixed point, we expect that the condition for the decorrelation that we derived for our linear network is fulfilled. And therefore, this prevents these tuning curves here from merging. However, when we stimulate at the peak here, indicated by this purple dashed line, then our network is strongly driven, and then we linearize the resulting network. The resulting linear network is probably no longer in the decorrelating regime, and therefore this leads these tuning curves to merge. So we are not 100% sure about this ourselves at the moment, but at least this is a potential explanation that can explain why these tuning curves here cluster and at the same time are decorrelated to some extent. And the question is, or the question we asked ourselves is this just some theoretical thing that is actually not important at all? So is there some usefulness into these distributions? And what we found is that one example where this might be relevant is hue perception, where a similar configuration of tuning curves here was hypothesized. And this may lead to the categorical perception of these different color hues. Because in such a configuration, it was shown that the response of the population vector here shown on the left to different color hues becomes clustered such that similar hues here can be easier classified by a linear decoder. And similar ideas have been put forward in other stimulus domains, as in, for example, auditory cortex, where it's about the perception of phonemes. And so more general, this principle of categorical perception might be another phenomenon that we can explain with our plasticity rules. Okay, with this, let me come to my summary. So what did I show to you today? First, I explained to you how we can learn co-tuned inhibitory and excitatory receptive fields with a modified Hebbian learning rule. When we apply these Hebbian learning rule in a recurrent network and we choose our parameters in a suitable range, we find that the tuning curves decorrelate and distribute across the stimulus space, while we observe like-to-like recurrent connectivity. And finally, our networks can perform useful computations that are learned exclusively from the feed-forward statistics of the inputs, and we have no hardwiring there. Okay, so as a last point, let me mention that inhibition stabilization and so inhibition stabilization in general and the SSN in particular are one of the major advances in theoretical neuroscience of the last decades, not only because the SSN can explain many experimental observations, far more than I touched today, but also because it's theoretically rather well understood. And therefore, we think that by adding plasticity to these types of networks, we made a huge step forward to actually understand how recurrent circuits compute and especially how they develop in the brain. Okay, and yeah, with that, let me thank my current supervisor, Juliana Georgieva, and the whole Max Planck group for their feedback and their input to this project. And of course, thank you all for your interest and for your time.